Now, I would like to invite our next speaker, who is basically the backbone of the gene therapy, as I may call, Andrea Perez. I think I'll for sure pronounce it wrong. Iturralde. The floor is yours.
Well, thank you so much for the introduction, Shivang, and I'd like to thank the organizers. Can you hear me? Sure. I really like to thank. Okay. So is this all right or not? Okay. I'd like to thank the organizers for inviting me today. And as it was already mentioned, I'm working at CMRI with Leszek, and we've been working on the development of an AAV-mediated gene therapy for CTNNB1 syndrome. I'm also gonna give a brief introduction, like a biology refresher, just so you are on the same page. And yes, DNA, the DNA is like the manual of instructions that all of our cells have about how to produce every protein in our body. So it contains all the information about us, from the color of our eyes, the shape of our hair, everything. And a gene will be like one of the chapters of this manual of instructions that will be responsible for the production of one of the proteins. So what happens in CTNNB1 syndrome is that one of the chapters, which is called CTNNB1 gene, has a typo, which we call mutation, and it leads to disease. CTNNB1 gene encodes for beta-catenin, and it has key functions in the maturation and development of the brain. On the one hand, it participates in cellular adhesions, where it mediates the communication between adjacent cells through the cadherin-to-cadherin interactions. And on the other hand, it also plays a key function in the Wnt signaling pathway, where in the presence of the Wnt ligand, it translocates into the nucleus and promotes the expression of genes that are involved in cell proliferation and cell determinations. And these functions are essential for synaptic formation and stabilization and for synaptic activity, so the communication between neurons. When there is a mutation in the CTNNB1 gene, these functions are also impaired. So the communication between the cells and the Wnt signaling pathway are not working as they should, and that's why there are symptoms in the patients. So our approach, as Leszek already mentioned, is to deliver a functional copy of the gene, and we use the adeno-associated viral vectors as the delivery box that will have the address where the gene should go in the body. So the cells will have the DNA with a mutation, but it will also have an additional copy of the gene that will compensate for the functions that are disrupted. And this is a representation of what will be happening in the cells. So the AAV vector will attach to the cells, and it will be internalized and go into the nucleus of the cell where the DNA is present. And here is where the AAV vector will release the vector DNA. It will be stabilized outside of the host DNA, and it will lead to the expression of the beta-catenin protein. Then the beta-catenin protein will exert its functions in the Wnt signaling pathway. It will again promote the expression of the genes involved in cell regulation, and it will also participate in the interactions between cells that are next to each other. So as no gene therapy for CTNNB1 gene syndrome had been previously designed before, we wanted to design different versions of the vector DNA or cassette. I'm gonna call it a cassette, expression cassette, in order to select the most optimal combination of elements that will lead to the compensation of beta-catenin functions. So as Leszek already introduced, we included in 6 different expression cassettes, the CTNNB1 gene, accompanied by different regulatory elements that were endogenous to the CTNNB1 gene, such as the 5' or the 3' UTR, or some intronic sequences. And the expression of this gene is regulated by elements that are already present in other gene therapy products that are in the market now in Zolgensma, such as the promoter and the poly-A termination signal. And all of these cassettes also included some sequences, the microRNA targeting sequences that will allow for the silencing of the expression of the gene in the liver and dorsal root ganglion. We want to avoid the expression in these tissues because it's been observed that the overexpression in the liver and dorsal root ganglion could lead to toxicity. So we tested these microRNA targeting sequences in a mouse model that was inoculated with an AAV9 that contained the expression cassette containing the green fluorescent protein as reporter gene, and it included or not this mixture of microRNA targeting sequences. And as you can see in the bottom panel, when we included these sequences, the expression of the green fluorescent protein was dramatically reduced. So we included these sequences in all of our cassettes that were individually packaged into the AAV vectors, and we selected the most optimal one in cortical brain organoids. These organoids were derived from patient cells that were collected and reset into a pluripotent state. This means that the cells are not blood cells anymore. They don't have any identity, and you can trigger their determination into any cell type that you are interested in, depending on the supplements you give to the cells. So due to the nature of the CTNNB1 syndrome, we were interested in neuronal cells. So we produce cortical brain organoids that will have all the different brain cell types. So after this, the first thing you need to know is that your model is actually representing what will happen in a CTNNB1 syndrome patient. So we wanted to see if there was differences in the expression of beta-catenin between a control cell line and a patient-derived organoid. And we could actually see the reduction in beta-catenin expression, both at the level of messenger RNA and at the protein level. We also wanted to analyze the functionality of beta-catenin, and for that we used two different markers that will indicate, will serve as indicators of synaptic activity and astrocyte presence due to the functions of beta-catenin in synaptic activity and astrocyte activation. And we could also see a dramatic reduction in the levels of these proteins. So we could confirm that these models could be representing what will be happening in the brain of the patients. So after that, we treated the organoids with the vectors that contain the different cassettes that we had designed, and we observed the normalization of the messenger RNA levels of beta-catenin with one of them. But we also wanted to test if we were also increasing or upregulating some oncogenic pathways due to the association of beta-catenin with tumor development. We could discard the upregulation of any oncogenic pathway, and we also closely looked at the Wnt signaling pathway. And these are the results that we obtained. So these are the different genes that participate in the Wnt signaling pathway, and the ones in blue represent the downregulated genes, and the ones in red, the upregulated ones. And you can see that the Wnt signaling pathway is still mostly downregulated, and many of these genes are actually effectors of the canonical Wnt signaling pathway that is associated with tumor development. And some of the genes that are upregulated are actually the inhibitors of this pathway. We also observed that some of the upregulated genes take part in another non-canonical Wnt signaling pathway, which is involved in synapse formation and neuronal plasticity. So this could be suggesting that there was some phenotypic correction in these organoids. We also confirmed this as we observed mostly an upregulation of the glutamatergic synapse pathway in these organoids. We also analyzed the protein levels. And again, you can see the differences between the control and the patient-derived organoids in the different markers. And after the treatment with the AAV containing the selected cassette, there was a recovery in the expression of these markers. So we selected this cassette for further analysis in a mouse model of CTNNB1 syndrome. And the cassette was packaged into an AAV9, and we inoculated the animals through the intracerebroventricular route with 3 increasing doses of the vector. We also assessed phenotypic correction. Pre-treatment, well, obviously, pre-treatment was like a control, a baseline, and we measured this correction 12 and 20 weeks, at 12 and 20 weeks of age of the animals. I'm gonna show you some of the results that we obtained. Firstly, we observed that there was a rescue in the anxiety-like behavior with the open field test. On the right-hand side, you can see that there is a phenotypic manifestation at 12 weeks of age, where we see differences between the control mice in blue and the CTNNB1 mice in orange. And we also observed a dose-dependent trend with the with the treated groups. These trends were not significant. However, at 20 weeks of age, we could actually see a correction with the highest dose of the vector. We also assessed the locomotor function with a kinematic gait analysis, in which you can monitor any movement, every movement of the animal. And we could actually see that there was a phenotype, a clear phenotype. I don't know if you are able to see that the CTNNB1 mice are slower, and also the hip and the tail are a bit lower. So the movement is also different. And we could actually extract different parameters from this monitoring. And on the right-hand side, you can see one of the parameters where we again see this phenotype manifestation at 20 weeks of age and the recovery at 20 weeks of with the highest vector dose. Dushko also analyzed the levels of expression of beta-catenin in these animals in different regions of the brain, and we could see a dose-dependent increase of the levels of beta-catenin. So that will give the explanation of why the highest vector dose group are having a correction in the phenotype. And we also observed that the levels in the liver were not different. So that could suggest that the microRNA targeting sequences that we include in our cassettes in order to avoid the expression of the gene in the liver are actually working. So this is kind of all the steps that we've taken. I've only shown you the results about the efficacy studies that we made in organoids and CTNNB1 mice, and Dushko and Ruth will talk about the biodistribution and toxicology assays. And for these biodistribution and toxicology assays, the vector needs to be produced under certain control conditions. So the vector was manufactured at ViralGen, where the clinical batch is also being, it was already produced. And it is important that we develop an assay in which we can demonstrate that the vector that is produced in ViralGen acts or works similarly to the vector that we produce in Australia and was able to lead to the increase of beta-catenin expression and function. So we developed a potency assay in which we use a common cell line that lacked beta-catenin expression. And in this panel, you can observe the beta-catenin protein expression. If you see a band, is that it means that the beta-catenin protein is being expressed. And if you don't see the band, it means that it doesn't express the beta-catenin protein. So we validated these cell lines and treated them with the vector produced in ViralGen. And we observed again a dose-dependent increase in the levels of beta-catenin, both at the messenger RNA levels and the protein expression levels. We also tested analyzed the expression of some target genes for CTNNB1 or beta-catenin, one of the genes that beta-catenin promotes expression. And in this case, we could also see a dose-dependent increase trend. So as a summary, we developed 6 CTNNB1 cassettes for CTNNB1 syndrome, and we could select one of them that had potential therapeutic effects without upregulating oncogenic pathways in cortical brain organoids. We further analyzed this cassette in a mouse model of CTNNB1 syndrome. We could observe a dose-dependent increase in the levels of beta-catenin and a phenotype correction. So these results suggest that the vector possesses therapeutic potential with a favorable safety profile. And finally, we tested the clinical grade vector, and we also observed an increased expression of beta-catenin in human cell lines. Finally, I'd like to thank all the collaborators, all the people in the lab who helped me, because this couldn't have been possible without every person who helped me one way or another. Of course, I'd like to thank the CTNNB1 Foundation and the Gene Therapy Research Institute, Spela, Dusko, Damian, and of course all of you, because you give us a purpose, and for me it's the most important thing. Thank you so much for your attention, and I'll be happy to answer any questions.